Okay. Mm, testing. Okay, I check my line. Okay, testing my mic. Okay, I think my mic has sounds. And then camera is okay, line is okay. Okay. So first of all, happy Merry Christmas today. So for those who are celebrating Christmas, happy Merry Christmas, and for those who are not celebrating, I wish you also Merry Christmas. So today, from your guys, you guys voting, you guys prefer want to have a class today. So I just respect your choices. You guys choice. You guys want to have a class, so I will have a class today lah. So today actually I will try to finish up the chapter 6 lah, photosynthesis. This is the last chapter. But for me it's not yet because I missed up chapter 4 yet. So after chapter 6 I will finish up chapter 4 lah. So this chapter, so actually last week I already explained about autotroph, right? The introduction of photosynthesis, and also give the how the photosynthesis work. So for today we will go toward the main point of the reaction. That is, as you guys know, photosynthesis have two reaction, right? Light dependent and light independent. So light dependent, another name is light reaction, right? And another light independent is dark reaction. So for today, I will proceed to this both reaction. So these two reaction are the very famous reaction lah. The most famous one. Everyone must master it and memorize it. Okay, so without further ado, so let's do. So normally light dependent reaction, it will involve light. Because he already say light dependent. Dependent means it depends on light. If got light, then it has the reaction. If no light at there, no reaction at all. That is why say it is light dependent because it depends on light. So normally this is the summary equation of the light independent reaction. So normally if you see here, if you notice that our photosynthesis will involve what reaction? Will involve water, right? The overall equation for photosynthesis is water and glucose molecule, right? Glucose plus glucose molecule to produce water and to produce oxygen and glucose molecule, right? Water molecule and carbon dioxide, and then will produce oxygen and glucose molecule, right? So these are the reaction in the light dependent. So it will only involve water molecule to produce oxygen. It only involves water and to produce oxygen. And then there are also the energy carrier. You can see that there are the two energy carrier that will be produced in this reaction, NADPH and also ADP. So NADPH actually is similar to NADH, just that it have the extra phosphate group at there. So if you still remember the structure of NADH that you learned in last chapter, chapter 5, you know that in aerobic respiration, the energy carrier produced, one of the energy carrier produced is NADH, right? But in photosynthesis chapter, no NADH is produced, it will produce NADPH. It contains the phosphate group. And then it also will produce ADP. So if you see here reaction, this is the overall reaction. It will use the water molecule and then the light energy to produce oxygen molecule and all this energy. So this energy, if you see that, if you notice that in overall of photosynthesis reaction does not have these two molecules, right? NADP and ADP does not have. Why? Because this is as an intermediate only. This energy will be used later by the dark reaction, light independent reaction. So that is why that if you write the overall photosynthesis equation, then you will eliminate your drop down these two element lah, two energy carrier because they are act as the intermediate only in the photosynthesis. Okay, so please remember that your light dependent reaction only involve water molecule, light energy to produce oxygen for the initial substrate and also final product. And then the intermediate is it will produce the energy. So the energy is NADPH and also ADP. So this is very important because this is one of the key difference between the light uh, dependent reaction and independent reaction. Okay, so in light dependent reaction, we have one key structure that play the role in this reaction. It is known as photosystem. So the structure of photosystem is look like this. So normally it is located at the thylakoid membrane. So the outside one is stroma, and then the inside one is interval of thylakoid. So it is located in the thylakoid membrane. It is located at the thylakoid membrane. So within the thylakoid membrane, we have this photosystem. So actually, this is the reaction central complex surrounded by light harvesting complex, meaning that there are many protein molecules we call as light harvesting complex and also reaction center complex. So actually, this complex is actually very important in the photosynthesis in the light-dependent reaction. So how they play the role? So normally reaction center complex, this one actually is an association of protein holding a special pair of chlorophyll A molecule and also primary electron acceptor. So this one actually is act as the primary electron acceptor. And then light harvesting complex consists of various pigment molecule bound to protein. So normally light harvesting complex, they will contain many chlorophyll pigment at here. So our chlorophyll A, chlorophyll B, and your xanthophyll, all these chlorophyll actually is located at the light harvesting complex. So the primary electron acceptor is a molecule that able to accept electron and becoming reduced. So normally, if you still remember that I teach you last week, you know that the photosynthesis reaction is it act like the chemistry, like atomic atom, the spectrometry that you learn in chemistry. You know that when the chlorophyll is absorb the energy from the light, right, the electron will become excited state, right? So if you see here, normally when the chlorophyll A at here, when it's absorb the energy from the light, then it will become excited state and then release the energy to another type of chlorophyll, another chlorophyll, and then this chlorophyll will be excited and then release energy to another chlorophyll again and then again and again until it reach the reaction center complex at here. So this is the main reaction will occur in the light independent reaction. So once the electron is already come through this chlorophyll A at the reaction center complex, after that the electron will be excited to the primary electron acceptor at here. So they are the primary electron acceptor. Later we will see it. What are they? So they will go to the electron will pass toward the primary electron acceptor, and then this electron acceptor will then go toward the electron transport chain. Dia akan pergi electron transport chain. So in photosynthesis, it also have the electron transport chain one, because it will produce energy. Dia nak produce energy. Kalau memang nak produce energy, memang akan ada satu system we call as electron transport chain. But there are some differences between aerobic respiration and photosynthesis lah. Okay, so this is the general key lah. Later we will see it very detailed. So normally in our photosynthesis, light dependent reaction, we have two type of photosystem, PS1 and PS2. So normally what are their difference? The difference is the absorption of the light. The absorption of the wavelength of the light in PS1 is 700. So what is known as P700. So meaning that the absorption of the light for PS1 is only normally is between 700 nanometer. But for PS2, it can absorb 680 nanometer. So this is the key difference. Another difference is their primary electrode acceptor. So normally in PS1, the primary electrode acceptor is ferrum sulfate, ferrum sulfur, FeS. Another one is Q protein. PS2 is Q protein. And then another one is, it will involve, this one is a very important one in the reaction. Either PS1 is involved both cyclic and non-cyclic, what involved as photophosphorylation. PS2 is involved only non-cyclic photophosphorylation. Okay, so what are they? So normally if you see, if you still remember this term, photophosphorylation, you know that phosphorylation is synthesis of ATP, right? Direct synthesis of ATP from the organic compound, organic phosphate. So this one, it add one term, what we call as photo. So meaning that this photophosphorylation, meaning it is special for the photosynthesis one. Only for photosynthesis only. Meaning it is also synthesis of ATP is coupled to the transport of electron that have been energized by photon of light. Because this phosphorylation is caused by the energy by the light. It is caused by the light energy. So that is why that it contains photophosphorylation. Okay. Okay. So now we go to the cyclic and non-cyclic photophosphorylation. So normally this one, if you see here, see this. So this is the arrangement of the PS system, photo system in the telecode membrane. So normally it is arranged like this. PS1 is at the right hand side and PS2 is the left hand side. So non-cyclic meaning that it is start from PS2. But for cyclic, you see that cyclic is start from PS1. And why it is known as cyclic? You will see here that when the primary electrode acceptor receive the electron, it will go to the FD and then it will pass through go to the electron transport chain. It will not go toward it here. You will see here, you will see the non-cyclic, it start from here and then it directly pass through and then I directly go to produce NADPH. But cyclic one, it is start from PS1 and then it will it will transfer back to ETC and then go back again. It is go back one. Cycle. That is why PS1 is only involve cycle. But for PS2, it will Right, that is why that we say as PS1 include non-cyclic and cyclic, because when in non-cyclic it start from PS2 and then go to the PS1 and then go to produce an ADPH. But for the cyclic one, it only involve PS1. PS2 does not involve. So meaning that when the electron go toward the primary electron acceptor, it will go to the ATC and then back to the PS1 again. It will not go to the PS2, and then this electron will also not go toward the produce an ADPH for cyclic. So this is the non-cyclic. So actually, if you see this diagram, non-cyclic actually is starting at PS2. So what I already say that the first reaction is that where the light energy will be absorbed by the chlorophyll A. Either chlorophyll A or B, but normally it's chlorophyll A. So when the chlorophyll A is already absorbed this energy, it will become excited state and then transfer the electron toward the chlorophyll. Dia akan transfer ke sini, ke sini. Will transfer to the another chlorophyll pigment. It will transfer to another chlorophyll pigment, another, and then until it reach the reaction center complex. Okay, once it reach the reaction center complex, the electron from the water molecule, so you will see here, water molecule, this is what we call as photolysis of water molecule. When the water molecule is already what we say is photolysis, meaning that it will release oxygen gas and also electron, right? So this electron will go toward here and then become excited toward this primary electron acceptor, what we call as FES. 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 So this electron, this is the primary electron acceptor. So this acceptor then will go toward the electron transport chain. It will go toward the electron transport chain. Why? Because it want to produce ATP molecule. So from here, it will produce the ATP molecule already. Okay, after that, this electron will go toward the P700. It will go toward the P700 and then it will undergo excited state again, and then transfer the electron toward the NDP plus reductase. So this enzyme is a key responsible to produce NADPH, to phosphorylate NADP plus to produce NADPH. So these are all the overall reaction inside the non-cyclic photophosphorylation. So in non-cyclic photophosphorylation, the main point is that it is starting from PS2 and then it will go toward ETC to produce ATP, and also it will go toward NAD plus reductase to produce NADPH. So there are three key points. And also the fourth point is it involves both photosystem. PS1 and PS2 are involved in this reaction. Okay. Okay, actually, this one is same what I mentioned. The image is same lah that before, but the more detail. You will see here there are the electron carrier at here. Excited reaction center, and there are the many reaction carrier at here. So okay, so if you see here, I already mentioned for PS2 the electron carrier is Q protein, right? What you will call as Q non-protein, and PS1 is ferredoxin, FD ferredoxin. This is the electron acceptor for PS1, and then the plastoquinone or quinone is electron acceptor for PS2. Okay. Okay. So normally it's like this. So okay. So normally this is the sentences. If you in essay, you need to write like this. So normally this one is I already mentioned. It's non-cyclic. It will involve ATP and NADPH production. So firstly, you see that the light energy is absorbed. The light energy, the chlorophyll will absorb the light energy and become higher excited state, so that the energy will be transmitted molecule until which the chlorophyll A of P has entered in reaction center complex. So normally the energy will be transmitted until it react it until it go to the until it reach the reaction center. Okay. Okay, and then you see here the energy will excite is called ACP. Okay, and then the electron in the chlorophyll A will be transferred from P680 plus to primary electron acceptor. On the other hand, photolysis of water include one enzyme split one water molecule into two electrons. So this electron will be supplied one by one to P680 plus once the electron in P680 plus transfer to the primary electron acceptor. This one is meaning that when the energy you see here, this is the energy. The chlorophyll A absorb the energy, absorb the light, meaning that the energy is transmitted until to here. So that when the electron is released toward the reaction center, the electron is released one by one. Once this electron become excited state, it will go toward this part. Go to the excited reaction center. You see here, this kuning yellow one this one is not excited yet, ground state. This one is excited state, and once it is excited state, it will release the electron to this electron acceptor, what we call as plastoquinone. And once it release, it will return back to the ground state. And then return back to the ground state when it have the energy and also electron, it will guide back toward the excited state. So this is the repeat process. So once it receive energy and also electron, it will become excited, and then it will release to PQ. And then once it release, it will go back to ground state again, and then receive the energy and electron again, and then repeat the process again and again. So that is what it means lah. These electrons are supplied one by one. It is one by one. Okay, and then you see here, there are also hydrogen ion. You see here, the photolysis of water will not only release electron, this electron will go toward the reaction center and also will produce oxygen. So this oxygen is our final product of photosynthesis lah. And another one is hydrogen ion. So this hydrogen ion actually will go toward the electron transport chain. So it will go toward the thalliquid space. So the theory is same as the chemiosmosis that you learn in chapter 5. Chapter 5. So it will go toward the thalliquid space and then undergo electron transport chain to produce ATP. Okay. Okay, so I think that's okay. Then it will go okay, this one is first part. Okay, later the photoexcited electron for primary electron excited of PS2 to PS1 by electron transport chain. So at the same time, light energy has been transferred by light harvesting complex pigment to PS1 reaction center complex. So the excite becomes P700 plus. The photoexcited electron is then transferred to the primary electron. So this one is same as the process. So at the same time, this PS1 also will absorb the chlorophyll A will also absorb the light energy at here, and then transfer the energy to this reaction center. So once this reaction center already received the electron from the electron transport chain, later it will directly undergo excited state and then release the electron to this ferrodosine. And then the electron come from the ETC that come from the PS2, it will go toward the NADP reductase. So that it will phosphorylate NADP plus to form an ADPH. So okay, so this is the reaction, the overall reaction of the non-cyclic. Okay, now we go to the cyclic one. Okay, we go to the cyclic. So the cyclic actually is more simple compared to non-cyclic lah. So actually cyclic is only involve PS1. And then after the similar reaction, if you want to write it, it is similar reaction. Just that the firstly also the light and the chlorophyll A and the PS1 also will absorb the energy. It will also absorb the light energy, and then this energy will be transferred to the reaction center. Just that once it receive the electron, it will become excited and then release the electron to ferrodosine. But this ferrodosine will go to the electron transport chain. It will not go toward the NADP reductase one. So you see this diagram, meaning that if the PS700 they receive the electron and then it will become excited state, and then this excited state will transfer electron to the FD ferrodosine. So this is the primary electron acceptor, and then this electron acceptor will transfer the electron to the electron transport chain. Okay, and then to produce ATP molecule, and then the electron will go back to the P700. So the difference is that the electron, the ferrodosine, will release the electron transport chain, the electron to electron transport chain, rather than release electron to the NADP reductase. So in cyclic photophosphorylation, it will only involve ATP production, and also it will not involve the photolysis of water one. Photolysis of water not one. So the energy come from the light, and then the electron is come from the electron transport chain. So it is a cycle. This is a cycle process. It is a cycle process. So once it go to here and then back to electron transport chain and then back to the C700. So meaning that the electron at here does not have the electron at here does not have the finish line. If you go to the PS2 one, the non-cyclic, it has the finish line. The final electron acceptor is NADP reductase. But for this one, it does not have the final electron acceptor one. The electron is recycled again and again. Again and again one. Okay, so this is the process. So normally, this is all the explanation. Actually, same as the PS1 only. Just that the difference is that I already mentioned that it will go toward the electron is passed from this one. It passed from the cytochrome complex for ETC to cytochrome complex for electron transport chain. And then the electron will pass back to the PS1 protein called plastocyanine, and then electron will be photoactivated again. So the electron will be photoactivated, the electron will be recycled, reused again. Okay, so that is what we call as recycle. So, okay, so normally the difference between cyclic and non-cyclic is already mentioned many times. I hope you can understand. Between this cyclic and non-cyclic is non-cyclic only it will involve both photosystem, and then but for the cyclic it only involve PS1 only. PS2 does not involve. This first difference. Second difference is that the production of energy. Both for the non-cyclic, it will produce ATP and also NADPH. But for PS1, it only produces ATP only. This second difference. Third difference is that the photolysis of water. Photolysis of water lah. Because it only non-cyclic will involve the photolysis of water, but for the cyclic, it will not involve. And also the electron acceptor for both systems are totally different. The PS2, the electron acceptor have two. Normally, plastocyanine and ferredoxin both are the electron acceptor, but for PS1, it only involves ferredoxin only. Plastocyanine does not involve. Okay, so I hope that everyone can understand it, but the theory is same. Just the electron flow is different only. Theory is same. But the first theory, the first reaction, light energy reaction are totally same. Just the electron flow is different. Okay. Okay, now we go to the chemiosmosis and electron transport chain in chloroplast. This is in chloroplast one, not in aerobic respiration, not in mitochondria. So actually chloroplast also have involved ETC. So what are their what are the what is okay, okay. So if you see here, so in ETC, if you still remember in chapter 5, the electron come from in chapter 5, chemiosmosis, oxidative phosphorylation, and electron transport chain that you learn in chapter 5. The electron is come from the energy carrier, right? NADH, right? Come from the NADH. NADH, FADH2. The energy is come from there. The electron is come from there. Right. But for here, the electron is directly come from here. The electron is come from this primary acceptor. So once the you see here PS2 and PS1. So once the electron they already PQ this plastocyanone electron acceptor, it will go toward this cytochrome complex. And then if you know that the water molecule, photolysis of water molecule also will release hydrogen ion at here. It will accumulate the hydrogen ion at the thalloid space. So it is similar, same as the chemiosmosis, right? But for the chemiosmosis, you still remember the chemiosmosis in aerobic respiration, your accumulation of hydrogen ion is at the intermembrane one. Right. You will still remember that in oxidative phosphorylation in aerobic respiration, your chemiosmosis is occur at the inner membrane of the mitochondria. This is on inner membrane of the mitochondria one. And then hydrogen ion is located is all of the accumulated at the outside one, at the intermembrane, and then inside the matrix is low hydrogen ion. But for here is reverse. You see here is reverse. The hydrogen ion is accumulated at the thalloid, and then it will produce the proton pump, meaning that the hydrogen ion for the reaction at here it will involve hydrogen ion can be come from two two sources. Either it is from the photolysis of water or in the electron transport chain. So in the electron transport chain in chloroplast, the hydrogen ion is pumped from stroma to the thalocode space. So it is a reversible theory from the oxidative phosphorylation one. Phosphorylation is you need to pump out to the intermembrane, but here is you need to pump in the hydrogen ion is pumped in inside the thalocode space. So inside the thalocode space, it already create the proton motive force, proton gradient, electrochemical gradient at here. So once it have the electrochemical gradient at here, meaning that this hydrogen ion will act as the energy and then to produce will pass toward the ATP synthase to produce ATP molecule. So actually the theory is same what you learn in chemiosmosis in chapter 5. Uh, so whatever you need. So if you still not understand what I say, please go back to the anti-transport, core transport, anti-transport, core transport. You should know lah for this. So normally if you see here what I say, energy surface reaction is energy and excited state for chlorophyll is transferred to the primary electron acceptor. And then after that, in the thalocode space, water will also release hydrogen ion. And then the gold arrow trade the flow of electrons. So normally the electron will flow from here to PS1. And then the 4 hydrogen ion are pumped into the thalocode space, increasing the so at the moment in the ETC will have the 4 hydrogen ion. One electron pass through will have the 4 hydrogen ion will be pumped into the thalocode space. So normally after that you still remember the concept of chemiosmosis. The diffusion of hydrogen ion for the thalocode space will back toward the stroma down the hydrogen ion concentration gradient through the ATP synthase. So this is what you learn in also same as the chemiosmosis in chapter 5, aerobic respiration. If you still remember your aerobic respiration, the chemiosmosis is pump the hydrogen ion from the intermembrane toward the matrix through the ATP synthase. But for this one, the location is different, different. The hydrogen ion is pumped from the thalocode space out to the stroma through the ATP synthase. So this is the what we call as when the hydrogen ion is pumped inside the thalocode space, we already create the electrochemical gradient. We already create the electrochemical gradient because it is low concentration to high concentration via anti-transport process. Okay, so I hope everyone is understand it. So normally this one is more detailed one. It will involve all the cytochrome complex. This one is PS2 and then this one is PS2 and then this one is PS1, right? PS1. This PS2, this PS1. And then this one is the cytochrome complex. Cytochrome complex. And then this one is NADPH, NADP betatase. And then you see here I already mentioned that inside the once the electron go to the receive the electron, it will provide the energy for the BEF to pump the proton ion inside the core. So this one we call as core transport. You call it actually transport, right? Actually transport process. It receives the energy and then this hydrogen ion will pump into the thalocode membrane. Same as the in mitochondria. Mitochondria is pumped to the intermembrane space. And then when they are pumped into this, it already create the proton motive force. Proton motive force, or we call as proton electrochemical gradient. So already create the proton gradient at here. So later, ADP, this hydrogen ion will pass through the ADP synthase to phosphorylate to form the ADP. So this one we call as photophosphorylation process. This ATP production is known as photophosphorylation. It is not oxidative, but it is photo. Why it is photo? Because the energy is come from the light. In in your ETC, in aerobic respiration for mitochondria, your energy, your electron energy is come from the oxidation process. It's come from the oxidation process. Your glucose, right? Glucose is oxidized, and then pyruvate is further oxidized. All the reaction is almost is all your energy is come from oxidation. So that is why it say oxidative. But for this the energy sources is come from the light. So it will not using oxidative, it will using photo. So in the ATP production here, you need to use photophosphorylation. Ha. Okay, so normally this one I just already explained it. You can be yourself. And another one lah, same what I mentioned right before, just another diagram. PS2, PS1, hydrogen ion, pump hydrogen ion here, and also there are hydrogen ion here. And then when the hydrogen ion accumulate at the thallicoid space, and then it will pump out to the stroma, to the ATP phase. Okay, so this one is the difference summary in cyclic and non-cyclic lah. So normally already mentioned many times lah. Photolysis already mentioned. What I've not mentioned wavelength, sources of electron. Yeah, sources of electron for cyclic is recycle. It recycle from PS1, but for non-cyclic it is from the photolysis of water. And oxygen, yeah, cyclic it will produce the oxygen because of eh already I think terbalik kot. Oh, wrong. Sorry. No. This one no. This one should be no photolysis. This one later I go back check. This one should be no photolysis one because I already said no photolysis. So meaning that no oxygen will be produced from cyclic. Only non-cyclic will produce oxygen. Sorry. Ever at here. Energy produced, yeah, it's like both have energy produced, but for cyclic, it only produce ATP. But non-cyclic, it only produce and produce both lah. And reduction, because reduction, this one also should be this cyclic is no, and then non-cyclic is yes. Terbalik. Take note for those who buy my note take note for this. And electron carrier, okay. So later I will edit it future lah. Okay. So I think that's all for light dependent reaction. So any question? Got question where manganese Mn complex on the thallicoid space, or what do you mean? You mean that the electron transport train occur at where? It occur at the thallicoid membrane. The electron transport train is occur at the thallicoid membrane. Here, thallicoid membrane. Inside is the thallicoid space, and then outside is stroma. So the electron transport train, if you want to ask, you ask the maybe like I said, ask you where is the electron transport train occur in chloroplast? It is occur at the thallicoid membrane. Same as what you mitochondria, right? Mitochondria is occur at the inner membrane, right? It is occur at inner membrane of the mitochondria. But for chloroplast, it is occur at the thallicoid membrane of the thallicoid membrane. It is occur in thallicoid membrane, not at the thallicoid space. You say thallicoid space is wrong. Thallicoid space just store hydrogen ion only. It is just to create the photochemical gradient. If you want to imagine as the oxidative phosphorylation mitochondria one, this one is the inner membrane. This membrane is the inner membrane, and then inside one is inside one is intermembrane, and then outside one is outside one is matrix. Terbalik. Outside one is matrix, and then inside one is inner membrane. For mitochondria case, if you want to imagine here, yeah, but actually it is the balik one, totally the balik. But the concept is still same, just the accumulation of hydrogen ion is different only. The accumulation of hydrogen ion for chloroplast or the thylakoid is inside thylakoid space. But for the mitochondria is outside, right? Is at the inner membrane. So dia terbalik je lah. Dia terbalik je. But the concept still same. Concept is still same. The chemiosmosis concept is still using the core transport that you learned in chapter 3. Core transport. I think I answered your question already. Okay, so any question yet for light dependent one? I know if no, then I will proceed to the light independent. No question. Okay. Now we go toward the light independent one. Light independent is the dark reaction that you learn in SPM. Dark reaction. So light independent, so meaning that you see the name independent, meaning it is not totally independent lah. Meaning totally independent meaning dia tak kisah dengan light cahaya lah. Got light or no light, it will go ahead. It will not depend the light lah. If for the light dependent reaction got light baru ada reaction. But for this tak kisah awak ada light, ada cahaya, tak ada cahaya, memang reaction akan terus. Unless dia ada carbon dioxide and energy je. For this one. Okay, so normally that independent reaction, the summary reaction is here using this. You will see here carbon dioxide and also water molecule. Right. Carbon dioxide, and also to produce glucose molecule. So and then you see that there are the energy carrier at here. I think it's 12. I crop the pictures of 12 NADPH. So this is the overall reaction. So meaning that it will involve the carbon dioxide. This reaction will involve carbon dioxide. It will not involve the light. And then it will use this carbon dioxide to produce glucose molecule. And then also will use the energy lah. This energy are produced from light dependent reaction. So what energy produced in light dependent reaction? It will be used in the dark reaction. So that is why that if you write the overall equation of photosynthesis, you will not write this. Because it is just as the intermediate only. It will be used later in this reaction. Okay, so in dark reaction, the most important one is carbon cycle. So this is another cycle that you need to learn lah. So normally this carbon cycle, another name is called what we call as carbon dioxide fixation. Carbon fixation occur in the soma. So normally dark reaction is occur at the soma. Light dependent reaction is occur at the liquid, right? But for this one is occur at the soma one. Soma spaces, the outside spaces lah. You see here, right? They chloroplasts, right? So outside the spaces is what we call as stroma lah. So normally this one carbon cycle, the main point is we need to fix the carbon dioxide. You know that carbon dioxide is very unstable gases actually. So normally you need to fix them. The plant will fix the carbon dioxide to become stable first. So normally this is the overall of carbon dioxide fixation process. So normally if you see here very complicated, right? Okay, we go to one by one. So normally this one what we call as carbon dioxide. So this one gray color is carbon, number of carbon. Any number of carbon at here. So you see here there are 3 circles, right? There are 3 circles, 3 gray circles at here, meaning there are 3 carbon. There are 3 carbon at here. So meaning that there are 3 molecules of carbon dioxide at here. 3 molecule of carbon dioxide. So there are 3 carbon dioxide enter this cycle. So in one cycle, it will only involve 3 carbon dioxide. It only 3 carbon dioxide first. It will involve 3 carbon dioxide first. So these 3 carbon dioxide will enter the carbon cycle through the enzyme that is very important enzyme we call as Rubisco. Very, very important. With no this enzyme, later we will see it. If this enzyme is absent, what happened to it? Later we will see it. Okay, Rubisco is very important enzyme. If no this enzyme, no carbon dioxide fixation occur at all. So this enzyme is very important. So it will fix with this molecule what we call as ribulose biphosphate. So there are 3 molecules of ribulose phosphate. So ribulose biphosphate actually is 1, 2, 3, 5 carbon molecule. So there are 5 carbon molecules here. Okay, and then so in easy word, each RUBP, each ribulose biphosphate will fix with each carbon dioxide. Satu RUBP akan fix satu carbon dioxide. Ini general. Just that what we say, why it contains 3, because of there are 3 carbon in one cycle, in every cycle will have directly 3 carbon dioxide enter it. So directly 3 carbon dioxide meaning that there are also require 3 RUBP to fix it. So once the 3 carbon dioxide is fixed with these 3 RUBP, it will form the short life intermediate lah. This is just a short life intermediate only. Short life intermediate. Wait. I think if I am not mistaken, it form short life intermediate, 6 carbon intermediate. Oh, okay. This one is it will form the short life intermediate only. Meaning that there are 6 carbon, right? You see here 1, 2, 3, 4, 5, 6. So you will still remember that this 5 carbon and then plus 1 carbon, it will form 6 carbon. No? 6 carbon. No? So this one intermediate, it will form the 3 intermediate 6 carbon molecule. This molecule is 6 carbon and they are total altogether are 3 6 carbon molecule at here. And then because it is intermediate, meaning it is very unstable, it will split up into 6 molecule of 3-phosphoglycerate. So meaning that this 3-phosphoglycerate only have 3 carbon only. So they are double to become 6 because this one is 6 carbon. So it will be when it becomes 3 carbon, meaning that it will double up. Karena ini 6 kan, ini ada 6 carbon. So kalau dia nak split into, dia akan berpecah. Kalau yang berpecah memang double lah. So double memang dia akan ada 6 will have the 6 molecule of 3-phosphoglycerate. And then it will use this one later. It will go toward it will undergo reduction process to dephosphorylate by using the ATP. So this one will use the 6 ATP lah. Because each molecule will use 1 ATP. So all together will have 6 ATP at here. So each molecule will use 1 ATP. So all together will have the 6 ATP and then to form the 1,3-biphosphoglycerate. So this one we call as phosphorylation process. 1,3-biphosphoglycerate. So another phosphate group is added at carbon number 1. And then after that, it will use the NADPH again. So another 6 NADPH they produce from the light-dependent reaction will be used by this molecule. So each molecule will use 1 NADPH to produce glyceraldehyde 3-phosphate G3P. Glyceraldehyde 3-phosphate. So this molecule later it will be split out, and one of the molecule will be split out to form glucose molecule, what we call as 3 carbon molecule. Meaning that if you all together got 6 molecule, right? 6 G3P. but only one molecule will be used to synthesize glucose molecule. But another other five will be recycled back to form ribulose biphosphate. So how to form back? Normally it will use another 3 ATP to form back this molecule. So altogether got 6 molecule, but only one molecule will be used in formation of glucose. Another other 5 will be recycled back to form the IUPP. So actually this reaction is very complex reaction, but in your syllabus in Campbell dia tak mention banyak lah. They just mention you will use ATP only. But if you refer to other book, dia biochemistry punya buku dia akan explain lebih detail lah. But I think because according to Campbell, dia tak explain very detailed, so you just want to know that it is regenerate lah. This G3P will be regenerated by using these 3 ATP molecule to form IUPP. So this IUPP later you will fix again 3 molecule of carbon dioxide and then repeat and repeat again. So if you want to produce one glucose molecule, you need have 2 cycle. 2 cycle. Nak 2 cycle baru boleh produce one glucose molecule. One cycle is not enough. Want to have 2 cycle. So that is why there some of the book is directly using 6 molecule of carbon dioxide. In Campbell, it used 3 molecule. But some book, many book, it will directly use 6 molecule. So you want to want wrong or correct? Actually not wrong. Yeah, it should be also correct. But just that in Campbell, it say that it use 2 cycle lah. But for here, it look like 1 cycle only lah. Because it directly use the 6 molecule of carbon dioxide. Just that they are double lah. You notice here, double 12, and then after that 12 molecule, and then got 2 molecule will be used for synthesize glucose molecule, and other 10 molecule will be regenerated to form IUPP. IUPP. So this is the overall process of carbene cycle. Okay, so normally more explained ini lah. This one is the chemical structure molecule. So the first reaction is carbon dioxide fixation. You still see here, this one more clear. See here, this is the IUPP structure. So the IUPP structure, actually the full name is ribulose 1,5-biphosphate. And then the enzyme, what I already say is ribulose rubisco, right? Actually the full name is ribulose biphosphate carboxylase. But in general, you can just use rubisco lah. Unless in structure question, unless it require you full name. Kalau full name memang awak kena full name lah. Okay. So you see here, this is more clear lah. The ribulose phosphate 6 ribulose phosphate will be fixed with 6 carbon dioxide to form this. 3-phosphoglyceride. Okay. Another one is reduction process. Okay, this 3-phosphoglyceride will use 12 ATP, so they are total altogether 12 3PG at here, and then will be used ATP to produce 1,3-biphosphate glyceride. So 1,3-biphosphate glyceride. So you see here each molecule of 3-phosphate glyceride will be 6-1-phosphate group through ATP from ATP through phosphorylation becoming, so this reaction is called as phosphorylation because it already received 1 phosphate group. Then there are total 6 molecule of this. This one I use Campbell one, but from here it is 12 molecule lah. Okay. There are only one out of 6 glyceride will be used for biosynthesis like glucose, and the other 5 molecule will be required to complete the cycle. So inilah. So if you want to synthesize one glucose molecule, you need 2 molecule of glyceride aldehyde 3-phosphate, meaning you need to have 2 round of cycle. Okay, so normally you will see here this phosphoglyceride, it will use using NADPH again, to form this glyceride aldehyde 3-phosphate. Okay, glyceride aldehyde 3-phosphate. Okay. And then after that you generate after that this but 1,3 biphosphor glycerol no glycerol diphosphate will be regenerated to form the rubisco again. To form the IUPP, no rubisco, IUPP again. So it will be generated to form IUPP again. Okay, so actually this one is in the biochemistry book in more detail lah. More detail. Actually, it's very detailed one. If you see Campbell just like this. 5 molecule of gazettia will be rearranged into 3 molecule of IUPP through phosphorylation by using 3 ATP. And ATP is phosphorylated to form phosphorylated to ATP in this process. So IUPP they can be fixed with another carbon dioxide and new cycle process. Okay, this is just Campbell. Tapi when you see in detail memang sangat detail. Let me see where are the first reaction? So kalau awak tengok Vibulose 5 phosphate Vibulose IUPP Vibulose 5 phosphate is the where okay, this one is our final, right? Vibulose biphosphate is our final product. Is the final product, right? This later it will go toward the carbon dioxide. So from here, actually, after the what we say another disaldehyde 3 phosphate is produced. Later it will undergo all these series of the reaction. So actually you don't need to know all these reaction. I just want you to show you the reaction is very complex actually. It's very complex. So it is not easy lah. Not say that it like directly regenerate. Actually there are many reaction here. Many reaction here. So you see here the reaction later it will form this DHAP, will form E4P, FBP, and then reaction banyak lah. It is very complicated reaction one. But for your syllabus, you don't need to worry it because Campbell pun tak cakap, so NPM pun tak akan cakap. You need to explain the three generation of IUPP. So you just want to know that this term lah. 5 molecule of gazettialdehyde 3-phosphate will be rearranged into 3 molecule of IUPP by using 3 ADP. You just want to know this. The others, if you interest, you can read it, but not compulsory lah. Just your extra knowledge for you. Okay, so normally if you see here, sorry. Sorry, apa lah. Okay, you see here normally there are 12 molecule, and then it will go split out into to form this DHAP and also form these erythrols, and then boleh tengok lah memang sangat complicated lah. Memang sangat complicated. So actually you don't need to know it lah. You don't need to know it. Tak perlu. Jangan risau. Memang tak akan keluar. Okay. So in this then these are the very important lah. In Karbon cycle, in one cycle, there are total 9 ATP and 6 NADPH are used in this process. This is only one cycle. So if you want to produce one glucose molecule, you need to double it lah. So total 18 ATP and 12 NADPH will be used to produce one glucose molecule. If you want to produce one glucose. But if you only want to use one cycle only, so one cycle only this 9 ATP and also 6 NADPH. Okay, so that's all for carbon cycle. So next I will go to photorespiration. So any question for carbon cycle? I think no question. Okay, so no question. I will go toward the photorespiration. Okay, so photorespiration. So this one, the carbon cycle process actually is on. Normally, you will know that photosynthesis is occur during the day, right? During the day, baru ada photosynthesis, and during night, no photosynthesis, right? In general concept that you learn in SPM, or even from kindergarten, primary school, but So actually, yeah, normally in general, photosynthesis is occur during the day, but there are some situations that during the day, the plant also cannot undergo photosynthesis. Why? Because of this photorespiration. So meaning that it occurs in the presence of light or other light, and as in aerobic respiration, it requires oxygen to produce carbon dioxide and water molecule. So meaning that actually in the reaction in the in the carbon cycle reaction, it should be the plant during the day should be fix the carbon dioxide to produce glucose. But when the plant have the photorespiration, meaning that light dependent reaction still occur, light dependent because got light. But light independent reaction is prohibited, inhibited. It is already inhibited. No light independent reaction. So meaning it will use oxygen and also produce carbon dioxide. So that is why they use photorespiration. Respiration meaning like human. Karena dia guna oxygen dan produce carbon dioxide and water same as human. But at the same time it also use light. Because it can use light to produce energy. So this term is that light dependent reaction function normally, but light independent reaction is already inhibited. So why it is happen? Because of this. Because our Rubisco enzyme, you see that this enzyme, this first enzyme, mana? Ni. This first enzyme actually is very sensitive to oxygen compared to carbon dioxide. Ha. So actually, this enzyme should be help carbon dioxide, should be fixed, should be help IUPP to fix the carbon dioxide. But this enzyme, unfortunately, actually in natural, it has a high affinity toward oxygen compared to carbon dioxide. So meaning that during the sunny day, if the day is very hot, normally the carbon dioxide concentration will be decreased. Why? Because of stomata. If you see here under hot average condition, leaf will lose water by evaporate through the opening in the leaf. Okay, and if like that, meaning that carbon dioxide from the atmosphere cannot enter to the plant. If you know the function of stomata, although you are not SEM 2, but in SPM you already learned. Function of stomata is what? Gases exchange, right? The main function of stomata is for what? Gases exchange. So meaning that our oxygen gas and also the carbon dioxide gas is passed through from here, from this stomata. So during very hot condition, yang senior awak tahu lah, during hot condition, your stomata is closed. So if it is closed, meaning that carbon dioxide from atmosphere cannot go into the plant, cannot go toward the leaf. So when carbon dioxide cannot go into the leaf, meaning the oxygen concentration is accumulated in the leaf lah. Maksudnya the partial pressure, the concentration of carbon dioxide in the leaf is decreased. So if it is decreased, previously oxygen increase lah. So when oxygen increase, memang Rubisco enzyme will bind toward oxygen rather than carbon dioxide. Also, this one will give the effect of photorespiration. So what are their effect? So apa effect there? The effect is when the IUPP is fixed with oxygen rather than the carbon dioxide, it will produce two molecules. One molecule is phosphoglycolate and another one is 3-phosphoglyceride. If you still remember that after when IUPP is fixed with one IUPP molecule, each IUPP molecule when we fix with carbon dioxide, it will produce 2,3-phosphoglyceride in normal situation. Each IUPP, when it fixed with carbon dioxide, it will produce 2,3-phosphoglyceride. But for the photorespiration process, it will not produce two 3-phosphoglycerate. It will only produce one, and another one is what we call as phosphoglycolate. Ini tak boleh lah. So when it produces the phosphoglycolate, this phosphoglycolate later it will be hydrolyzed to glycolate, so it will use water, and then this glycolate will use oxygen to produce glycine, and then this glycine will produce glycerate, and then to produce carbon dioxide. So meaning that the concentration of 3-phosphoglycerate is already decreased. If it already decreased, meaning that there are not enough reaction to continue the carbon cycle. So you know that 1,3,3-phosphoglycerate later it will use ATP, right? Dia akan guna ATP to produce 1,3,5-phosphoglycerate, right? But for now, because of the it already kurang satu, should be if you think that if there are 3 IUPP, should be produce 6 3-phosphoglycerate. But for now, it only produce 3 phosphoglycerate, and another 3 is phosphoglycolate. 3 IUPP 3 IUPP in general dia akan produce 6 3-phosphoglycerate, 6 molecule of 3-phosphoglycerate. But for photorespiration, because of oxygen, dia akan hanya produce 3 phosphoglycerate only. Another 3 is forming phosphoglycolate. So meaning that the 3-phosphoglycerate is already known, the concentration is already decreased. It cannot go toward the carbon cycle again. Kerana tak cukup lah. Kan? And then this phosphoglycolate later it will use water, and then it will use oxygen, and then to produce carbon dioxide again. So this is what we call as photorespiration. So dia macam human, but just that it will use light during the day, and then respiration like human. Kerana dia ambil oxygen and produce carbon dioxide. That is why that we call as respiration. Okay. So this is what we call as photorespiration. So in the chloroplastoma, yang ni, I don't know why the image is they blur the image, but I just put it lah. So normally this one what we call they assume that this is chloroplast lah. They chloroplast. So the chloroplast IUPP will be add oxygen, and then glycolate is formed. And then after that, this glycolate will be diffused into peroxide, where it is converted to glycine. So what here lah, converted to glycine. And then glycine will move toward the mitochondria and is converted to serine again. To release carbon dioxide. And then this serine will move back to the perimysium to convert it to glyceride. And then the glyceride will move to the chloroplast, and where it is converted to 3PG and enter the carbon cycle. So ini cycle dia lah, ini the whole reaction lah actually. This one is more easy. Actually in glycine, awak tengok kan, dia glycine dia cakap dia akan move to the mitochondria. You think that inside the chloroplast, IUPP will be added with oxygen. This one is occur in chloroplast. Once the glycolate is produced, it will diffuse into the peroxide, where it is converted into glycine. Okay, and then the glycine later will move to the mitochondria and converted to another amino acid called serine, and then this time it will release carbon dioxide. And then this serine will back toward the peroxide again, and then to form the glyceride. This is our final product lah, glyceride. And then this glyceride will later recycle back to the chloroplast, and then it will be converted to form 3PG again. 3PG again. So dia akan then go toward the carbon cycle. So you see the overall process. Yeah, glyceride finally it will go back to the chloroplast, and then to form the phosphoglycerate, and then so that the phosphoglycerate can produce to the carbon cycle. So if like that, the carbon cycle is not meaningful already, because it will already produce carbon dioxide. Dia produce carbon dioxide. Macam lah ini tak logik lah. Memang tak logik lah. Kalau awak nak produce carbon dioxide, you want to use I can say that if photorespiration, it already reduce the rate of photosynthesis. Kerana dia kurangkan reaction of the carbon cycle. Dia ada halang lah. Tak boleh buat smooth pun lah. Kalau macam lah highway. Kalau ada highway, kalau carbon cycle itu highway, memang 3-phosphor-glyceride it can directly go. But for now, because of photorespiration, dia macam jalan lama lah. Ada lampu isyarat, jalan dia kecil-kecil, dah pusing-pusing ni, pusing sana, pusing sini punya. Kan? Yang pusing sana, lepas tu pusing balik, form back again. Go to the highway again. Like when you want to Penang go to KL, go highway, right? So in carbon cycle is the pathway you directly go. But for the photorespiration, you use the awak guna jalan lama. Jalan lama awak pusing lah, pusing sini, pusing sana, pusing balik. Tapi finally awak balik ke highway lagi. You wasting your time. So it reduce the reaction and reduce the productivity of the glucose molecule lah. So this is the explanation. So normally this photorespiration is occur in C3 plant, what we call as C3 plant. C3 plant. So if C3 plant like that, so in some plant they can overcome it. How to overcome it? So there are another plant we call as C4 plant, which is the leaf of the C4 plant is Kranz anatomy structure. You will see this is the what we call as Kranz anatomy structure. So if you see this diagram, our diagram D, you will see here the structure of the C4 plant is total. The palisade mesophyll cell is rounded. Dia macam surrounding to the vascular tissue one. Surrounded toward the vascular tissue one. But for the C3 plant, no. C3 plant is like the normal plant that you see in your SPM lah, the structure of the leaf. Okay. So normally C4 plant has this structure, what we call as the there are two type of it will involve two type of cell. So one type of cell is mesophyll cell. This one it will involve mesophyll cell, and then another cell we will use is bundle shift cell. There are dual cell here. So if in C3 plant it will not have this one. Bundle shift cell tak ada. Dia hanya ada mesophyll cell and then directly go to the vascular tissue. You will still remember the normal structure of the plant, normal structure of the the normal structure of the C3 plant is like this. It's like this. This is the vascular tissue. Betul ke? Vascular tissue. And then the mesophyll cell, it also have bundle shift cell. Sorry, C3 and C4 both contain bundle shift cell. Just the arrangement of the leaf is different. The mesophyll cell is located at here. It is located loosely at here. But for the C4 one, you see here, the mesophyll cell is located surrounding punya. Dia memang surrounding punya. Tengok dia macam circle kan. This one what we call as Kranz anatomy lah. Kranz anatomy structure. Dia macam bulat-bulat macam ni. And then bundle shift cell is at the is tightly packed to the bundle shift cell. But for this one, mesophyll cell for C3 plant, the bundle shift cell does not take over in the carbon cycle one. It just located outside from the vascular tissue. Mesophyll cell then arrange loosely lah, arrange ikut suka hati mereka nak lah. But for this C4 plant, the mesophyll cell is arranged bulat-bulat macam ni punya. And then the bundle shift cell pun bulat-bulat macam ni punya. And then because bundle shift cell, it will surrounded to the vascular tissue. So it surrounded like this. Just the mesophyll cell is arranged in Kranz anatomy structure like this. So in C3 plant, only mesophyll cell involve light independent variation. Light independent variation for C3 plant, it only involve mesophyll cell. Bundle shift cell tak involve. Dia tak involve punya. But for C4, they are involved. Mesophyll cell and bundle shift cell are involved in the light independent variation. Okay. What are their difference? You see here, mesophyll cell carbon dioxide fixation and the carbon cycle are totally occur inside the mesophyll cell. But for C4 one, the carbon dioxide fixation is occur at mesophyll cell first. The carbon dioxide is fixed to the mesophyll cell first. Then after that it will form the intermediate what we call as C4. That is why we call as C4 plant, because the first intermediate that enter the carbon cycle contain the molecule is carbon 4 molecule. It contain 4 carbon. But for carbon cycle, the carbon cycle the molecule enter the carbon cycle is carbon dioxide and it contains 3 molecule. Right, 3 molecule of carbon dioxide. This is why we call as C3. Karena ada 3 molecule of carbon dioxide enter. So this total 3 carbon, right? So this is C3. But for this one, the molecule that enter the carbon cycle is contain 4 carbon. So that is why we call as C4. So this C4, it will form carbon dioxide is fixed in the mesophyll cell first, and then directly to form the C4, what is called as oxaloacetate. If you are not mistaken, oxaloacetate. Later I will go to it. Oxaloacetate, and then after that it will go to the fixed form the carbon dioxide again, and then fix to the carbon cycle. So there are that dual process. So okay, so normally, this one is more detailed. You see here, the carbon dioxide it will enter the mesophyll cell, just that the carbon the enzyme used and the carbon that fix this carbon dioxide is different. It is not Rubisco lah, bukan Rubisco. Not the IUPP lah, not IUPP. It will involve what you call as PEP. PEP. It will involve PEP. So PEP, if I'm not mistaken, is phosphoenolpyruvate. Phosphoenolpyruvate were used to fix the carbon dioxide. And then once it fixes the carbon dioxide, it will produce oxaloacetate. This is the 4 carbon. So this is the reason why it use C4. Karena dia produce 4 carbon. For C3, it produce 3 carbon, right? Ini produce 4 carbon. So that is why it says C4. Ini nama dia macam ni. C4. So after that, this oxaloacetate later will be oxidized to form malate. Okay, and then this malate later will enter the bundle shift cell. It will enter the bundle shift cell. So this bundle shift cell, malate will be split into 2 molecules. One of the carbon molecule will be released as carbon dioxide. It will release carbon dioxide, and then another 3 carbon will form pyruvate. And then this pyruvate later will be entered again to mesophyll cell to form back the PEP. And then this carbon dioxide later it will release toward the carbon cycle, and another carbon dioxide fixation occur. So in C4 plant, it will have 2 carbon dioxide fixation process. First carbon dioxide process is at mesophyll cell. It will use for PEP carboxylase. Another carbon dioxide fixation is occur at carbon cycle. Dia ada dual carbon dioxide fixation. So this one can prevent that because this PEP carboxylase is more affinity toward carbon dioxide. So that it will not bind to the oxygen one. So directly carbon dioxide can directly bind to the PEP using this enzyme. But if no this enzyme, no this structure, dia macam ni lah. Kalau dia terlalu panas, carbon dioxide concentration too low, and it will bind to oxygen. IUPP will bind to oxygen. So it is the carbon cycle still maintain, just it have the extra carbon dioxide fixation process. Carbon cycle sama je saya cakap tadi, just that it contain another carbon dioxide fixation at the mesophyll cell. So the key differences is it contain 2 carbon dioxide fixation process, and then the carbon cycle is occur at bundle shift cell. For C3 plant, it occur at the mesophyll cell. And then this lah. Okay. Yang ni pun sama lah. Okay. Okay, so normally this is all the explanation. So the most important is that you need to know that mesophyll cell is the first carbon dioxide fixation process occurred here, and then it will directly produce oxaloacetate and then be reduced, oxidized, reduced to malate, and then this malate will enter the bundle shift cell. After that, this malate will be oxidized to form pyruvate and release one carbon dioxide molecule. So actually, this one is what we call as the oxidative decarboxylation. Oxidative decarboxylation process. We call it oxidize and then release carbon dioxide. And then this carbon dioxide later will be fixed again into the carbon cycle. So Rubisco will be take part again. So Rubisco still take part. This process, just the main point is that because under the hot sunny day, PEP can play the role to bind carbon dioxide, but IUPP cannot because IUPP will bind to oxygen during the hot sunny day. Okay, so this is the difference. So ini perbezaan dia. So I already cakap banyak kali lah. So this one first product. So normally C3 plant, the first product is 3-phosphoglyceride. First product for C4 is oxaloacetate. Because of this is a key point kenapa kami cakap C3 and C4. Because phosphoglyceride is 3 carbon, oxaloacetate is 4 carbon. Based on the first product lah. They naming the plant based on the first product. And then the carbon dioxide acceptor for C3 only IUPP. For C4 contain 2 lah, IUPP and PEP lah. Okay. So I think and then we have another plant we call as CAM plant. CAM. Okay. Apa dia perbezaan dia? So normally CAM plant is something look like C4. It will also, you see here, it will also carbon dioxide will be fixed to the C3 and then to form C4, form oxaloacetate, and then after that it will convert back to the oxidize back to the carbon dioxide, and then the carbon dioxide will enter carbon cycle to produce glucose molecule. Same as the pathway, the reaction, same as the C4, but the difference is it does not contain Quantz anatomy structure one. This CAM plant does not contain Quantz anatomy structure one. It will only involve mesophyll cell only, same as the C3. The reaction only occur in mesophyll cell, but it can use the reaction pathway that's similar to C4. So what are the key difference? Apa perbezaan dia? Kenapa dia boleh? Kenapa C3 tak boleh? Kenapa CAM boleh? Kerana mereka guna satu sel kan? Kenapa CAM boleh why? Because the reaction, the time occur in this reaction is different. This reaction, the first carbon dioxide fixation will occur only in night. In C3 and C4, both C3 totally occur during sunny day. C4 also occur during sunny day. C4 plant, both carbon dioxide fixation, all of this process is all occur only during sunny day. Night, no this process one. Night, no this process. But for this one, they are pecah. The first carbon dioxide fixation occur at night. After that, dia baru second carbon dioxide fixation baru occur during the day. Meaning that you see here, normally the first reaction is still same, just that the malate produced from the oxaloacetate, malate produced will be stored in the vacuum first. Yeah, simpan dekat vacuum first. It will not directly go toward carbon cycle one. Ini C3 tak ada. Kerana C3 dia directly using Rubisco to fix the carbon dioxide. So if during sunny day memang dia tak ada mechanism macam ni. But CAM plant have. because if CNM plant, if because it only involve one mesophyll cell. So kalau lah dia occur macam C3 punya. So walaupun dia ada odaloacetate C4 punya mechanism pathway, but no meaning because until finally you also will produce carbon dioxide, but Rubisco is high affinity toward oxygen, so you are not solving the problem actually. So no, so if you want to have this reaction, this reaction help him, the first carbon dioxide fixation will only occur in night. So during masa malam, dia plant CAM plant akan absorb lah semua, will absorb all the carbon dioxide, akan absorb all the carbon dioxide and store in the vacuum, and then to form the bind to the PEP, and then form a dialoacetate. After that, reduce to malate. After that, it will store. This malate will not directly oxidize to form carbon dioxide. It will store in the vacuum first. So this is due to the prevent of the pH of cytoplasm decrease due to malate is acidic. So why this malate want to store at the vacuum? Because want to maintain the pH value in the plant lah. In the plant. If not, kalau malate terlalu banyak, it will decrease the pH value of the plant. So it will store in the vacuum first. And then during the day, they already have energy. ATP and ATPase is already produced, so they already have energy. This malate will be diffused out from the vacuum. And then after that, this malate will be oxidized to form pyruvate and remove carbon dioxide. And then so that this carbon dioxide will be used again to the into the carbon cycle. So ini maksudnya. This one is just that we want to increase the carbon dioxide concentration in the plant. During the hot sunny day, they just want to maintain the carbon dioxide concentration high inside the plant. Kalau kerana dia ada store many malate in the vacuum, meaning that walaupun in hot sunny day the stomata is closed, carbon dioxide cannot enter, but inside the plant still have many carbon dioxide here, so that the Rubisco still can bind toward the carbon dioxide rather than oxygen. So that is why that they use this mechanism. Dia macam simpan dulu. Kalau nak guna, baru guna. Tapi C3 tak ada. C3 no this mechanism, but CAM have. Walaupun dia hanya guna satu cell. So that's for CAM. So this is the difference between C4 and CAM. So the mechanism pathway totally same. Just difference is cell. C4 2 cell. CM only one cell. Mesophyll cell only. And then the carbon dioxide fixation first and second is different. First is occur during at night. Second is during at day. But for C4 both are at day during the day. So ini C4 and CAM punya difference. So you can make yourself. Okay. And then this one is also very important because nowadays the MPM is very want to use the example. Because for nowadays they will not use in essay question lah. They nowadays MPM very like to use the plant name, and then want you to write out the mechanism process, meaning that if you don't know that time the plant example of the C3, C4 and CAM, you don't know which pathway. Is it C3 pathway, C4 or CAM pathway? So if you need to also not just learn all these variation or C4, CAM, okay, tahu. Nanti dia keluar example, awak tak tahu. Nowadays MPM dia suka macam tu lah. Dia tak akan keluar directly cakap C3, C4. No lah. Dia akan macam main awak guna cakap Bali. Yeah. Write out the production or ATP and ADPH of Bali or carbon cycle of Bali or pathway of Bali lah, like that lah. Awak kena tahu lah. Bali is under C3. So these are all the example of C3. So normally C3 plant yang ni lah semua. C4 punya ini C4. The most important, the most famous one is these two lah. maize and sugarcane. Maize and sugarcane is very famous one. The other you can also find it. CAN, the most important is pineapple and cactus. Pineapple and cactus is very famous one, and also orchid. Orchid, and also other snake plant, kalanchoe, moringa, sedum, all these lah. Okay, so that's all for the light independent variation. So any question? Let me see. No question. Okay. If no question, okay, next we go to the last topic lah. So, okay, so this is the last topic, limiting factor. Okay, this topic, I know everyone is very bored. You think very boring. Very bored. But I want to say that got 1 year, I think this year only, or I don't know is last year or last 2 year, it come out in essay question. Two times essay question. Dua kali lah. Saya ingat dua kali lah. Yang satu is this compensation point. Another one, if I no mistaken, is about carbon dioxide. Dia dah keluar dua kali lah. So if you think that you don't want think that it is not important because dia akan keluar normally is B lah. A is about the carbon dioxide, about the pathway. Lepas tu B is limiting factor. 5 markah, I think 6 or 5. You know how to answer A. A, wah, boleh lah, jawab. Tapi B tak tahu. Kalau B, awak tak baca. Because I know many people don't read this. Even your school teacher will not teach you also, I think. Even at my time, I study, I also don't read this. But luckily lah, that time I don't cut out in essay. But unfortunately, your senior lah, now same 3 senior, I think they come out this question. What kind of question is this compensation point? This one that actually you already learn in SPM. Dia keluar dari SPM punya. MPN dia keluar dari SPM punya. Memang semua SPM, memang dia SPM topik punya. SPM topik punya. So because I think this SPM you already learn, so I don't know you still remember or forget. You forget, okay, I'll teach you again. So normally there are the point what we call as the compensation point. So you know that compensation point is the level of the light intensity at which no gain or loss of sugar amount occur in the blood. Meaning that this is the point, this is the point that the light intensity point that no photosynthesis, the rate of photosynthesis and the rate of respiration is equal. No sugar is used, no sugar is produced. Equilibrium. What is say that is equilibrium? Apa itu equilibrium? Chemistry, please go back. Refer. Apa itu equilibrium? Meaning that the rate of photosynthesis and then rate of respiration process is equal. No product is formed, no sugar is used. The glucose molecule, the use of glucose molecule for respiration and the production of glucose molecule for photosynthesis is similar. And then also other molecule lah, semua sama aja. It is equilibrium process. What is said as compensation point. So this compensation point is based on the light intensity. So if the light intensity is below than the compensation point, so compensation point meaning that respiration rate is more higher. At lower light intensity, no photosynthesis. Even though no photosynthesis and the respiration rate is very high. Why no photosynthesis? Because no energy. Because no energy, the light energy is not enough. Tak ada light. Because you need to know that light dependent variation, the photosynthesis, the first variation is light dependent variation. Dia nak energy. The energy is come from light. If no light, memang tak boleh lah. So no ATP and NADPH4, so Krebs cycle also cannot proceed it. But when the light intensity increase, increase higher than the compensation point, rate of photosynthesis will increase also. And when the light intensity reach the compensation point, the rate of photorespiration and photosynthesis is equal at the time. And then if light intensity continue, it will increase the production and pH, and then the chlorophyll pigment have become saturated. And then higher than saturation point, there is no further increase of rate of photosynthesis. So this is because light intensity now is not the limiting factor but other factor. So meaning that although the light intensity you can increase the rate of photosynthesis, but there are also the saturation point, same as the enzyme concentration punya topik. Oh, saya tak belajar lagi, tapi I think your school is already teach it. So dia akan become saturated. Dia akan reach the maximum lah. Kalau chlorophyll pigment, the each plant have the limited chlorophyll pigment. Chlorophyll pigment memang kalau light energy terlalu kuat, walaupun terlalu kuat, dia akan reach maximum. Walaupun tak kisah apa kuat, chlorophyll tak boleh absorb lah. Kan? Hanya boleh absorb macam ni je. Tak boleh lah. Kan? Tak boleh absorb lain lah. Ini masuk dia lah. Okay. So that's all for compensation point. So actually this one is very easy lah. So actually it's very easy. You will know how to answer it, but I don't know because you guys don't focus. So keluar semua orang tak tahu. Another one is about carbon dioxide. So actually same as you think logic pun tahu lah. If you know that the carbon dioxide used in carbon dioxide fixation, so normally what are the function of carbon dioxide because of carbon dioxide fixation? Okay, so normally at low concentration of carbon dioxide, your rate of photosynthesis will be very low, will be decreased, very low. And then the low level will act as the limiting factor of photosynthesis. And then in C3 plant, low carbon dioxide concentration will cause photorespiration. Okay, and then yang ni kalau dia carbon dioxide increase, photosynthesis will be increased. And then but it will also have the saturation point. So if higher than saturation point, no further increase. See, because light intensity now is not the limiting factor, but other factor. So if you want to increase the photosynthesis, maybe you need to rearrange the other factor. Same as here, same as the light intensity. Because light intensity now, if you higher than saturation point, meaning that it is not a limiting factor, you need to, if you want to further increase the rate of photosynthesis, you need to modify other factor. Okay, so carbon dioxide normally you can use the theory that you learn in C3, C4, and CL. You can, awak boleh tulis but you need to according to this lah. Another one is temperature. So this is the last one. So normally, temperature also they affect one. So normally low temperature and extreme high temperature will act as a limiting factor. If too low or too high, it will cause rate of photosynthesis is very low. So if too low, like winter day, you see that during winter day, the winter country like United States, China, Japan, winter day, the tree does not have leaf, right? Tree pokok dia tak ada daun punya. Memang hanya tebang je. Hanya only have the stem only. Because no photosynthesis, because too cold. But if too high, if kalau terlalu panas, memanglah enzyme tak boleh function and also even can burn out. Forest fire lah, it can cause forest fire. So normally the optimum temperature it need to based on the plant also, but normally optimum temperature is 35, 36 like that lah. But compared you need to based on the location. If like desert can up to very high temperature lah. Okay. So I think that's all for this chapter, temperature. So ini limiting factor, you don't need to think it's very difficult. Think logically. It just logic punya. Tak susah pun. Kalau dia keluar memang senang je. Tak susah punya. Think logically you can write it. Okay, I think that's all for class today. So any questions? So I already finish this chapter. So any question for this chapter? So I think no. Okay, so if no, so tomorrow I will enter the last chapter lah, chapter 4, enzyme. So okay, tomorrow I will enter it. No, hopefully next Monday is New Year. 1st January, 1st January. So 1st January I will complete the all the syllabus in semester 1 lah. Yeah, if you want to buy the note, you can screen the I have post the link lah. Let me copy the link. Yeah, if you want to buy the note, please buy through this link. Yeah, buy through this link. I don't know you can access or not. I think cannot. Uh, yeah, I think this link, this latest link. If you want to buy the law, you just access this link. You also can access my group channel. My group channel I already also got post the link also. Okay, so any question? Any question again? I think the last two I don't know why they are macam keluar. You use the latest one, the only link one. I think that's all. So tomorrow I will finish up the. I don't know I can finish it up. I think I cannot finish it up tomorrow. Chapter 4. I think need to until next Monday. So hopefully you guys can understand lah apa yang saya tak ajar. Okay, I think that's all for today class. So thank you for your attending. Although it's holiday, you guys still attend my class. So whatever. So happy Merry Christmas. So and also good day and good night for all of you. So thank you. Bye. See you tomorrow.